So today I will be doing a photosynthesis review. The majority of the information in this review comes from BioNinja, me just synthesizing and restating, analyzing the stuff on BioNinja, and a little bit comes from some articles off the internet. So photosynthesis is the process in which cells make organic compounds like glucose from inorganic molecules. So the goal is to synthesize organic compounds from inorganic compounds. It starts with 6 carbon dioxides, 12 waters, and then using light energy and chlorophyll pigment produces a glucose, 6 oxygen, and 6 waters. So like during cellular respiration in animals, chemiosmosis is used in an electron transport chain to make ATP. This ATP can be used by the plant directly for active transport, growing, and more, or it can be used to make other organic compounds. Just like in animals, the glucose or organic compounds produced are used in the mitochondria in cellular respiration. Photosynthesis requires light energy and a photosynthetic pigment. When we look at plants, we see the color green, meaning plants use all the wavelengths except green. So the reason I'm outside is to show some real life examples of this. So this is my magnolia tree. She's really beautiful, and obviously you can see the leaves are a beautiful green color. There we go. And so once again, the reason why this plant is green is because it uses every wavelength of light except the green, because that's what's reflected. On the electromagnetic spectrum, visible light is in a range of 400 to 700 nanometers per wavelength, with violet, the shortest, being 400, and the longest being 700 nanometers with red. Chlorophyll, the plant's photosynthetic pigment, absorbs blue and red light the most strongly and reflects green light. I remember this by the fact that I've literally never seen a blue plant. So I have this handy diagram right here. You can see that green is used the least and blue, like right up here, is used the most. Red is used quite a bit over here as well, and then it tapers off when it leaves the spectrum of visible light. When the Earth was first formed, there was little oxygen in the atmosphere. It was an anoxic environment. However, oxygen started accumulating because photosynthetic organisms produced it. It was first produced in the oceans because that was where the first life was. But once all the iron in the ocean reacted with the oxygen to form iron oxide, oxygen began accumulating in the atmosphere. So here's another diagram to show that. Is how the oxygen levels at first were little to none, and then it began increasing, peaking, and then decreasing. So and no O2 is produced. It starts to fill the ocean, and then it accumulates in the atmosphere. So actually, this is kind of an interesting fact. 2.5 million years ago, most of the bacteria living in the oceans were anaerobic. The cyanobacteria developed and flourished, producing large amounts of oxygen, which is what the process I said earlier with the oxygen making iron oxide in the oceans and then entering the atmosphere. This oxygen was actually toxic to most forms of life on the Earth, which is weird to think about because we think of oxygen as giving life. And so the number of anaerobic bacteria dropped significantly. Also, the O2 combined with methane to make CO2, leading to a rapid cooling of the planet. There was a mass glaciation, and most all life on Earth perished. Even the cyanobacteria were threatened. So this event is called the Oxygen Holocaust, or the Great Oxygenation event. So to me that's just very interesting because at a time oxygen was toxic because it was new to the environment. However, by now everything's adapted, and as you can see by all the green plants around me reflected in the mirror, in the glass, that oxygen has become a staple of all life on Earth today. except for some anaerobic bacteria still exists, but only in the depths of the ocean where there's little to no oxygen. Okay, so now on to the big stuff. Photosynthesis itself. Photosynthesis can be split up into two processes, the light-dependent reactions and the light-independent reactions. Firstly, light-dependent. The end purpose of the light-dependent reactions is to convert light energy into chemical energy, namely ATP and NADPH. This occurs in the membrane in between the two membranes of the thylakoid. So step 1. The photosystems become excited by light energy. So this green thing right here is a photosynthetic pigment, or a photosystem. So they're organized, so photosynthetic pigments are organized into groups called photosystems. When light hits it there, the electrons become excited. These electrons are transferred onto carrier molecules in the membrane itself. So you can see that right here, the electrons become excited, then they're transferred into the membrane itself. So step 2, the production of ATP via the electron transport chain. As the electrons move along the membrane, they lose energy by splitting water into H+ ions. H+ ions enter the inner, so this is the inner membrane space inside the little tiny thylakoid, green disc. So the H+ ions are transported into this membrane space. The thylakoid is very adapted for this function actually because the inner membrane space is so small. The H+ ion concentrations build up very rapidly. So a concentration gradient is formed with many H+ ions being right there and few being outside the membrane there. The H+ ions rush through ATP synthase, which is an enzyme in the membrane, which converts ADP into ATP. So ATP is formed through phosphorylation. To break down this word, photophosphorylation. Photo means light, phospho referring to phosphate in ADP and ATP, and ation indicating a process. So as the H+ ions rush through to escape the concentration gradient, they form ATP through phosphorylation. Hey, you can see a reflection. My camera's kind of broken. So step 3, reducing NADP+ and the photolysis of water. So as electrons moved from photosystem 2 to photosystem 1, which you actually can't see in that diagram, they only gave up some of their energy to splitting water into H+ ions. These electrons are transferred onto carrier molecules, NAD+, which forms NADPH. This is called reduction. So NADP+ is reduced to NADPH. Alright, so that's all the light-dependent reactions. In conclusion, ATP was formed, and NADP+ was reduced into NADPH. So the light-independent reactions. These are a cycle with the goal of producing glucose. This cycle is called the Calvin cycle, and conceptually it's just the reverse of the Krebs cycle from cellular respiration. So this cycle occurs in the chloroplast fluid called the stroma. Step 1 is carbon fixation. Fixation means converting atmospheric CO2, so just the gas everywhere, converting it into a combined form like, well, in this case, CO2 joins a 5-carbon molecule called RUBP. This RUBP becomes a 6-carbon molecule with the carbon dioxide that was fixated. This molecule is very unstable, so immediately splits into 2 3-carbon molecules called G3Ps. In step 2, G3P is reduced. G3P is converted into triose phosphate using NADPH and ATP. So G3P becomes triose phosphate, or TP. In the final step, RUBP is regenerated. 6 TPs are produced per cycle, and one of these is used to form half a glucose. The remaining 5 are turned back into RUBPs for the cycle to restart again. So because RUBP starts the cycle, and then 5 TPs are cycled to remake the RUBP. So it takes 2 full Calvin cycles to produce 1 molecule of glucose, but this is still a fairly efficient process, as 1 molecule of glucose results in about 30 ATPs, and then only about, I can't remember, maybe 12 ATPs are used in the Calvin cycle. So it's kind of lengthy, and only half of glucose is a little sad to think about with all the steps I just listed, but it's still fairly efficient, and that's why plants are very adapted and great. Okay, so that's my photosynthesis review. Once again, most of this was BioNinja and the biology textbook, and then also the little bit of stuff about the oxygen holocaust came from an article on it.